Good afternoon, all of you. In this class, I want to discuss about selection and screening of recombinant cells. While discussing about different types of vectors, there we have already discussed how the recombinants are screened. If you remember, while discussing about the plasmid vectors, for the screening of recombinant cells, certain characteristics had been incorporated in those vectors so that we would be able to screen the recombinants. Say, for example, while discussing about the vector PBR322, twin antibiotic resistance was the property that had been introduced in those vectors so that enable us to screen the recombinant cells. Always it has been a challenge to distinguish this recombinant cells, that means the transformed bacteria containing your recombinant plasmid from the transformed bacteria containing the unaltered vector. I hope you are able to understand what I mean. Transformed cells are the ones that take up the external DNA. During this process of recombinant DNA technology, the external DNA could be the recombinant DNA, or it could be the unaltered vector, which is the major problem during the process of the production of recombinant DNA. But we have certain methods that will enable us to screen these recombinant cells from the uh, the transformed cells containing the unchanged or unaltered vector. So now let us see what are all these procedures by which we would be able to select these recombinant cells. This is also the topic given in unit 2. Though all the procedures are studied while discussing about different classes of vectors, under this heading, how do we explain the different methods of screening these recombinant cells? Yeah. The procedures for direct selection of recombinant cells, there are three different ways by which we can screen these recombinant cells. One is by using reporter genes or marker genes. Second, we should be able to eliminate the non-transformed cells. When we allow the transformation reaction to take place, it's not that all the cells will be transformed. You will have the mixture of transformed and non-transformed cells. So you should be able to eliminate the non-transformed cells after the transformation reaction. Then we also have methods for the identification of clones having the recombinant DNA. Direct methods are also available. Now let us see one by one how do we use these methods to select the recombinant cells. So these reporter genes are nothing but marker genes. They produce a particular phenotype. This produced phenotype by this marker or reporter gene that permits the easy selection or the quick identification of the cell. So this selectable marker, or I mean this reporter gene or marker gene, that could be either selectable marker or scorable marker. We have seen several selectable markers already. Whatever we have been studying the selectable marker, those are antibiotic resistance genes. So antibiotics like kanamycin, they're all selectable markers. So when a population of bacterial cells, when they are placed on the medium containing certain antibiotic like kanamycin, only the cells that have kanamycin resistance gene only will survive. The remaining cells will not be able to survive. This we know very well. This is how we do antibiotic selection, which is nothing but the selection using a reporter gene or marker gene. Whereas scorable markers, they also produce distinct phenotypes, but based on these phenotypes, we would be able to identify these recombinant cells quickly. Examples of such scorable markers are beta-galacto-urinidase, luciferase, octopine synthase, nopalin synthase, and green fluorescent protein. So these are all the genes which produce such enzymes are the scorable markers. They produce certain distinct phenotypes, and these phenotypes allow the easy identification of the recombinant cells. Then how do we eliminate these non-transformed cells? Obviously, these non-transformed cells can be eliminated because of the presence of the selectable or reporter genes. And generally, the vectors, whatever we develop, a good vector has generally two marker genes. Uh, Among those two marker genes, one will be the selectable marker. Say, for example, if you take PBR322 vector, plasmid vector, it has got two selectable markers, tetracycline and ampicillin resistance gene. So, the non-transformed cells in the reaction mixture, that means in the transformation reaction mixture, can be eliminated by plating them on a medium containing the selection agent. So here the selection agent could be either tetracycline or ampicillin. That is in case of the plasmid vectors. We also have studied phage vectors, bacteriophage vectors, cosmid vectors or phagemid vectors. In all those vectors, after the recombinant DNA molecule is produced, they are packaged into a virus particle, and these virus particles will be used for infection of the bacterial cells. And these infected cells will be then plated on the bacterial lawn. If you remember, all this we have studied while studying about bacteriophage vectors, lambda replacement vectors we have studied there. How the procedure of selection of recombinants is done, we studied in detail. The same thing I am repeating here. I hope you are able to recollect all those points. So what I am saying, in case of phage vectors, cosmid and phagemid vectors, after the recombinant DNA molecule is produced, they are allowed to be packaged into virus particles, and these viruses will be used for infection of bacterial cells. These infected cells are then plated on susceptible bacterial lawn, where they would be able to form plaques. You all know how the plaques will be formed because of the repeated cycles of lysis and reinfection. You will see the formation of plaques. So these plaques, as you all know, are formed due to the vector infected cells. But of course, these plaques would have formed either by containing the recombinant DNA or unchanged vector. It doesn't matter whether it is recombinant DNA or unchanged vector. Because of the virus particle, the plaques are formed. So further, you need to have a procedure to screen the recombinants. So the third one, which will allow us to directly screen the recombinant cells, that is by using direct antibiotic resistance screening or blue-white color screening. I hope you remember, just now I mentioned this direct antibiotic resistance screening is nothing but having two antibiotic selectable markers on one vector, so that you can go for insertional inactivation of one particular marker gene. Similarly, blue-white color screening also, the LAC-Z gene is used for insertional inactivation, by which you would be able to select the recombinants directly. These two methods are extensively discussed when we were discussing about plasmid vectors. If you remember, the twin antibiotic resistance property is found in the PBR322 plasmid vector, whereas blue-white color screening is found in PUC series vectors. It can be PUC 8, 18, 19, all these vectors, they contain this blue-white color screening property for the selection of recombinant cells. So I will not go through these points. Let me go to this diagrammatic representation and then recollect all those points quickly. So if the plasmid has only one selectable marker, that will help us to select the transformed cells. That will only help us to select the transformed cell. So here in this picture, what it is shown, there are cells having the chromosomal DNA. There is no plasmid. Probably this is the host organism. We select. Then we have our vector. If it is a plasmid vector, in which we would be introducing our gene of interest. And usually a plasmid vector should contain a selectable marker. In this particular example, the selectable marker shown is ampicillin resistance gene. When we allow the transformation to take place, obviously cells will take up the external DNA. So you will have the cells containing this external DNA. But here there are two different types of cells. Here the plasmid without gene of interest. Always there is possibility in the transformation. Your gene of interest gets incorporated in the plasmid, which is the desired molecule, our recombinant molecule. Whereas it can also get religated without the incorporation of your gene of interest. So that is the religated vector or unchanged vector. So this is the most possible side reaction that can take place during the ligation. So when you allow the transformation reaction to take place, either this molecule can get inside the cell or this molecule can get inside the cell. But this is the molecule we are interested in screening. This is the molecule containing our recombinant DNA molecule. But in the reaction mixture, the transformation reaction mixture, you will have the combination of these two cells. So these cells also will be present, these cells also will be present. So to see, our challenge is to eliminate these cells and then screen these cells. But there is, when there is only one selection marker in the vector, we can only eliminate the non-transformed cells. Only we can eliminate these cells. The cells that did not take up the external DNA can be eliminated. So if you allow this mixture to be plated on the plate containing the ampicillin, that will allow the growth of the transformed cells, but that does not help in screening the recombinants. So that is the reason we go for thin antibiotic resistance, where the vector contains selectable markers, two selectable markers that confer resistance to two different antibiotics. So this diagram is not new for you. Already I have discussed while discussing about these plasmid vectors. Screening of recombinants by insertional inactivation of a resistance gene. As I told you, they will contain two resistance genes. Here in this example, PBR322, TET-A gene is also there, ampicillin resistance gene is also there. So when you incorporate your gene of interest, that is incorporated in the coding sequence of one of the selectable marker, in this example, that is in the coding sequence of tetracycline. So when this is incorporated in the coding sequence of tetracycline, what will happen? The recombinant molecule is produced. Always there is possibility, as I mentioned, without the incorporation of your gene of interest, that can get religated to form the unchanged vector. So after these two products are formed during the ligation, you have to go for the selection of these recombinant cells. There, if you remember the method that we use for selection of recombinants. What is the method for selection of recombinants by this method? One of you please answer. I am waiting. Are you there? Students, where are you? I am proceeding. I did not get any answer from you. I will talk about it later. I am proceeding further. Okay, always you will come up with excuses like my mic is not working. Respond in chat box. I want to listen from you.
Yes, ma'am.
Who is that?
Shweta, ma'am.
Shweta, I have asked a question, right?
Yes, ma'am.
So what is the answer for the question I asked? When we go for this insertional inactivation of a selectable marker, what is the method we use for selection of recombinants? This is not new for you. Earlier we discussed. You don't know the answer, Shweta?
Ma'am, blue-white screening.
No, it is not blue-white screening. Blue-white screening is the one when we insertionally inactivate the LAC-Z gene. Here I am talking about thin antibiotic resistance. Okay. Anyway, I'll proceed further. Please follow it. So here to select the recombinants, what we do is we use the method called replica plating. So here, first we allow the cells to grow on a medium containing ampicillin. So when we allow the cells to grow on ampicillin, both, I mean here it enables the selection of transformants, meaning the recombinant the cells containing recombinant DNA also grow here, and the cells containing unaltered vector also will be growing here because both the cells have the intact ampicillin. That's how both the colonies, that means all the transformed cells will grow here. Then we do the replica plating technique. We take a nitrocellulose membrane and or absorbent paper. We just take the imprint of these colonies and then transfer onto the another plate containing the medium. Along with the medium, it also contains ampicillin and tetracycline. So when that medium contains both ampicillin and tetracycline, it allows the growth of only those cells containing the intact tetracycline resistance gene. That means it allows the growth of only those cells containing unaltered vector. So from this plate, we can mark the positions of those colonies containing the recombinant plasmid. And from the master plate, you would be able to pick up the colonies containing recombinant DNA. So this is the replica plating technique that can be used to select the recombinants. If the vector what we select consists of two selectable markers. Then the next method which we see in case of Puck series vectors is blue-white color screening. This is another method for the selection of recombinants. So I will directly go to the diagrammatic representation because already all this procedure is discussed in detail. This is just the repetition, but people can take it as revision and then understand this once again. But here in this topic, this is the information you have to represent. So here what we do, there is a LAC-Z gene. This LAC-Z gene sequence is insertionally inactivated by incorporation of your foreign DNA. Here also there is possibility for the recombinant plasmid and unaltered vector to be formed. When we allow the transformation to take place, both the DNA, in the sense unaltered vector or the recombinant DNA, can be taken up by the bacterium, but you have to go for the selection. For the selection, we use the plate containing the substrate X-gal, and also IPTG is added in the plate. Why IPTG? If the host organism is producing the LAC repressor, in that case, we use the IPTG for the induction. So these are the components that are present in the selection plate. What are all the components present in the selection plate? So here the steps are given. Foreign DNA are both cut with the same restriction enzyme. Of course, then only there will be compatibility. Then it is inserted into the plasmid where it inactivates the LAC-Z gene. So if the LAC-Z gene is intact, it produces the enzyme beta-galactosidase. That beta-galactosidase that converts X-gal into the blue colored product. In case of recombinant plasmid, since it insertionally inactivates the LAC-Z gene, there is no beta-galactosidase produced. When there is no beta-galactosidase produced, no blue colored product will be formed. So when we allow the cells to grow in this plate containing IPTG and X-gal, you will see the combination of white colonies and also the blue colonies. You all know that the blue colony is the non-recombinant, the cell containing non-recombinant DNA. Why? Because there is no, there is no beta-galactosidase. When there is no beta-galactosidase, what will happen? Agar HCl plus IPTG. When there is no beta-galactosidase is synthesized, the X-gal will be converted into blue product. When there is beta-galactosidase is not produced, then the X-gal will be will not be converted into blue product. So that's how we would be able to differentiate the recombinants from the non-recombinants. I hope you guys are able to follow. In case of the non-recombinant, black Z gene is intact. It produces beta-galactosidase, wherein X-gal is converted into blue product. Whereas in case of recombinant, since there is no intact black Z gene, there is no beta-galactosidase produced. When there is no beta-galactosidase, there is no conversion of X-gal into blue product. So such colonies will appear as white colony. So white colony indicates the cells containing the recombinant plasmid. So this is how when we do this experiment, the white colonies and blue colonies appear on the plate. So these are the techniques which we use for the selection of recombinants. In addition, we also discussed few methods to select the gene of interest from the gene libraries. So those also had been discussed in detail. Several methods are there, but two important methods are hybridization, nucleic acid hybridization, and immunological screening or immunological staining. These are the two important methods which are used for the selection of gene of interest from the gene library. So these are also the methods of screening recombinants. So this is how the nucleic acid hybridization is carried out. We have the master plate where we plate the colonies of bacteria containing recombinant plasmid. That means the clones from the DNA library will be plated on the master plate. After the colonies are grown, you use nitrocellulose filter paper and take the imprint of these colonies. When you take the imprint of these colonies, then you treat this membrane to lyse the bacterial cells. When the bacterial cells are lysed, the DNA will be released. Then you allow the DNA to be denatured so that the double-stranded DNA will be denatured to single-stranded DNA. After this treatment of this nitrocellulose filter paper, you allow the radioactively labeled DNA probes to bind to the DNA present on this nitrocellulose filter paper. So if it finds its complementarity, the probe will go and bind to the DNA. So here you would see the colonies containing plasmid of interest because of the binding of the probe. So here it is shown in close up. The gene of interest, if it finds the complementarity with the probe will go and bind there. And since the probe is radioactively labeled, we can expose this filter to photographic film so that the position of the desired colonies can be detected by autoradiography. So based on this position, the corresponding colonies can be picked up from the master plate and it can be analyzed for the further characterization. So that's how the nucleic acid hybridization is also used for screening of recombinants, which is the method commonly used for the selection or screening of gene libraries. Similarly, we also have immunological screening. So here what we do in immunological screening, the same principle recently we discussed when we were discussing about Western blotting. So here you digest that with eco R1, then you allow your gene of interest to be incorporated, that will produce a fusion protein, and in vitro packaging can be done, plate on the bacterial lawn. This we are discussing in case of a bacteriophage vector. So when you allow the in vitro packaging and if you plate it on bacterial lawn, plaques will be formed. Then you can use the nitrocellulose filter paper to take the imprint, then remove the filter paper. Then the proteins are bind to the nitrocellulose filter here. The bound proteins should be detected by immunological screening. Here first what you do is you can incubate the filter with primary antibody, specifically the primary antibody will go and bind to the protein of interest, then wash the filter, then you can incubate the filter with the radioactively labeled secondary antibody. Then what will happen, since the secondary antibody is radioactively labeled, you can expose it to the X-ray film and detect the presence of these colonies, I mean the plaques containing your recombinant DNA. So this is immunological screening. So these are all the different methods which are used for the selection of recombinant cells. So this is all I wanted to discuss in today's class. If you have any questions, you can ask me. This is also the topic in second unit. Many times this question had been asked for 15 marks also, or sometimes it has been asked for 5 marks also. Either way, you should be able to answer this question. Any questions?